Hi everyone. I'd like to welcome you to the 9th Fenix Infrastructure webinar. This is the 9th webinar in our webinar series. And I'm Nicoleta Chiapidou from the Barcelona Supercomputing Center. I will be the host for today. Our webinar today is called Using the ICEI Resources for Atomistic Molecular Dynamics Simulations and COVID-19 Example. And our speaker for today is Modesto Orozco from IRB Barcelona. I would just like to let you know that we're recording this webinar so that we can post it online on our website afterwards, along with Modesto's presentation slides. And the structure of the webinar is as follows. After this very short introduction, Modesto will have his presentation, and then we will have a Q&A session so that you can ask any questions you have or make any comments. You can use the chat at the bottom right of the Zoom feature, or you can click on raise hand so that I can unmute you and you can ask your question out loud. And in the end, I'll just wrap up the webinar with a few concluding slides. So I think we can begin. Modesto, I'll give the floor to you. I'll stop sharing now my screen, and you can share your slides.
Okay, so can you hear me, all of you? Hope so.
Yes, yes, yes.
Well, so
all good.
Thank you for the invitation. It's always a pleasure, to help to spread the importance of high performance computing. And so today I'm gonna make a kind of very general talk about what exaflop and big data. So what big computers can do to increase our knowledge about COVID-19. So what I'm going to explain today is not only my work, so there is a part of the work of my group, but I have put in a bigger context, in the context of the BioExcel Centre of Excellence. So I'm going to explain about results from many different groups. I'll try to make clear all the time who is the owners of the results. But let me start with some general conclusions. Let me start with things that I have learned. And in this crazy year, so it's about 1 year that the COVID arrived to Europe, and actually it has blocked our life. It's incredible. So if you put back a year ago, you will never predict what has happened. No? And in this year, where I think science has done very well, a few conclusions I think are very clear. The first is that time matters. So I have problems in my projects in my group that have been running for, 10 years. COVID projects are always urgent. So you cannot work in this paradigm of slow science, everything well done. Time is not an issue. Here, time is an issue. Second and third are more or less the same. I mean, we cannot work anymore in the isolate paradigm. We need to integrate efforts and we need to share information. And so I'm not a paranoid, but if we can do, I mean, we means my group can do something by itself. I typically don't look for collaboration, so I try to do by myself. And of course, I share information, but rarely I share raw data because you think that, okay, in your data maybe something else, and you don't really want to give advantage to your competitors. So this is the standard paradigm, or this has been the standard paradigm. With COVID, all this has changed. And these are other things that I have learned. The first, that high performance computer is really useful. So there is a lot of criticism in my field, whether these big centers, these big computers are really useful, whether they are kind of dinosaurs that implies a lot of resources and that are not returning what they cost. I'll try to convince you that yes, they are really useful. Second is that Center of Excellence are really useful. I think you will be convinced that without the Center of Excellence I'm talking about, without BioExcel, all that I'm going to show you today won't be possible. And finally, and this is probably my most important take-home message, is that computer scientists are useful. So when actually work together with people that know the problem, they can really make a difference. And I will convince you on this point also. Okay. So when the pandemic started, as I told you, we have already set up and we were working together for several years, the BioExcel High Performance Computer Center of Excellence. So it's an initiative that was created to help people doing simulations. And actually, as everything was set up, we were able to react very quickly. You know, without a center of excellence, this won't be possible. But as we already have done many infrastructure together, it was possible to put this infrastructure, change gear, and instead of, I don't know, studying other things, focus all of the technology into COVID. And one of the first things that was done was an initiative from KTH that was the first open data repository for COVID-19. So it includes data of everything. And one of the interesting databases that it contains were structural databases of all the proteins that are important in COVID, all the COVID-coded proteins. And we actually, this is a database that is growing continuously and is growing, for example, in terms of molecular dynamics simulations. All the big groups are putting the trajectories there. But then we decide in Barcelona to do something a little bit extra. Instead of just, giving out trajectories to this central data repository, we decide to take the data from the repository and actually add value. So the idea was that we had already developed, and this was in the context of BioExcel and also in the context of this consortia with Jülich and other centers, that is the Human Brain Project. And we decided to take all the infrastructure that we have developed for human brain targets to the COVID problematics, and then build a website that you can actually visit. It's open. This is we have a working version, but this is the public version. And in this place, we add a lot of extra things to the trajectory, so that an external group can take a look at the trajectory and see really the information that he's expecting or he or she is expecting to find. So it means that in addition to the trajectories, we introduce a bunch of data. So this is this is an ultra short video. So we have this kind of data, we have trajectories, we have one data, we have all kind of metrics of deviation. We have a bunch of, for example, context, how the interface changes, flexibility, all kind of flexibility measures, local, global, principal components, boundaries, hydrogen bond, interaction surface, important contacts, interaction contacts for drugs, cavity searching, etc., etc. And this is a very fast video. Let me show you examples. This is, for example, the virus polymerase. Is the central machinery for replication of the virus. This is a crystal structure that we and many others have simulated. And you can see, for example, here, the pattern of hydrogen bonds along the trajectory, what is stable, what is not. This is the RBD here at the right, SA2 domain. So these are the interactions between the virus and the human protein, that this is the required step for viral internalization. And for example, here you can, in this case, you can plot what is the boundary of interaction in AC2. And this is very useful to understand how the RBD, how the virus is recognized, and what will be the impact of any mutations in the human protein. Or this is another example. This is a search for cavity. Honestly, I don't remember which was the target, but you can see all the druggable cavities, so all the places where, if you are working in a pharma company and want to design a ligand for this complex, you should take a look and do docking. And this, I give you here a flat image, a rigid image. In reality, this is an ensemble, or many other things. For example, this is it was from a trajectory from the show, where actually they were looking at ligand. I don't know if you can see the mouse. This is a ligand. It's a potential inhibitor of this protein, and then you can trace how all the residues in the protein interact. For example, you realize immediately that this is, I think is, yeah, I think this is an aspartic. Aspartic 78 is crucial for interaction. And then you can guess, okay, this is very important, but take care. If the virus mutate here, the drug is more likely to be inactive, and then you can actually trace residue by residue, and they will have a dynamic picture of the interaction. And then you can go do many things. You can connect to all the databases that are around, and you can do things that, I mean, I think are very interesting. For example, what you have here in the bottom is spike, that is a central protein. So if you are an expert, you will recognize this is one, this two domain, and you will recognize the RBD. And what you have here in green and red, green, orange, and red, are mutational entropies. What it traces here are those residues that have been mutated, and those positions that have been mutated by the virus with higher frequency. So the virus has been with us for a year. It has a lot of time to mutate. And then you can see that actually most places in a spike, in the external part of a spike, have been mutated already. And you can actually look at these images in a dynamic point of view, see how accessible they are, how they are not, whether they are glycan protected or not, and you can compare, for example, with epitopes. So these are the regions which are recognized by the antibodies. So these are the vaccine targets, and then you can compare whether these mutations are gonna fit in a place which is important for epitope definition and actually may somehow hamper a part of the vaccine response. So we have all this data there, and you are absolutely free to put your data there, to actually analyze the data that is there. You are also very welcome. You have trajectories to tell us what type of analysis you have, because the analyses are customized for the type of protein. So this is a completely open data initiative, and I might say that all the very big groups in the field are actually very keenly giving us the trajectories. Okay, but in BioExcel, we did many more things. For example, we adapt. So more than develop, we adapt HPC tools, and this is an effort where many people has been working. This is the work of Alex von Ben in Utrecht, where he actually refined Haddock, uh, both Haddock server to meet the increased needs, and also the core of the program to actually adapt and make it more efficient for docking exercise. And I will show you later a few results that he got. And also big effort from different partners, the Manchester, BSC, Elixir, with ourselves were involved in trying to develop a kind of workflow hub. So a place where people deposit it and actually use workflows that can be useful in COVID research. I will comment with a greater detail one of these workflows in a while. But we have also, as the groups in BioExcel, we have been quite active doing simulations ourselves. So trying what I label as modeling studies. So trying to learn how the virus work, how can we interfere the viral replication cycle, etc. And this is I start with a massive study from Alex Bomben group, where he actually do a massive docking experiment against RNA-dependent RNA polymerase. This is the central machine that takes and duplicates the RNA. And then he did a docking, and these are the highest score. And quite amazingly, quite nice, remdesivir appears as the first hit. So it's based on his results, it looks like remdesivir will be a nice binder for polymerase. We know that it's a polymerase inhibitor. So we start with this point and then start working and actually start with very similar to Alex's study. We take a look to remdesivir binding and the binding of the different oligonucleotides to the enzyme. And it was I would say, quite amazing finding that was apparently a very well refined enzyme. And why I say it's a surprise, because come on, this enzyme is in a virus that has a few years of life, probably 2-3 years. So it's a very new virus. And even the coronavirus family is it's supposed to be from the Roman time. So it's a couple thousand years. So it's a new organism compared with us that we have millions of years of evolution. So despite this novelty in the planet, it has a very good ability to recognize. You can see here how well adapt to the shape of the incoming nucleotide. So it means that it's going to be a high affinity and um, high accuracy replication. In any case, we use these structures to start a little bit different study. In this case, was a KMMN study. So the objective was, okay, we are binding remdesivir, but what happened later? So it's just a non-covalent or is a covalent and inhibitor. So in other words, is incorporated into the RNA or is just blocking the progression of the RNA replication in the binding site. So we did then some quantum mechanical calculation. And in this way, we learned how the enzyme works. It was another surprise. So let me show you this video. You can see here, this is the template. So I'm sorry, the template is behind. This is the 3', and here you see the incoming nucleotide. You see here how the phosphate is broken. This is PPI that will be left. And then you have the reaction. This is amazing because it's exactly the same mechanism that you will find in human polymerases. And remember, you are talking about a virus that in the best case has a couple thousand years of evolution refinement compared to our cells that have at least 1 million years of evolution refinement. But then the barrier that you get is about, for a normal oligo, is about 15 kcal, and this is more or less the same barrier of our polymerase. But then if you repeat the same simulation with remdesivir, you see that the barrier increases just a little bit. What is this strongly suggesting it is actually remdesivir is not a pure non-covalent inhibitor, it's actually something that is incorporated into the RNA, into the nascent RNA. Okay, let me jump very quickly to other works. In this case, I will comment on a very nice set of studies done by KTH in collaboration with other groups. And this is at that time was the one of the main targets for drug design. That was the main protease, trying to get ideas about the dynamics and how, as you can see here, you can actually fit inhibitors in this protein. Also, this on the very similar lines is the work of the people in Finland looking at the spike protein. You see here the wonderful spike protein, and they were interested in trying to understand this opening movement, the exposure of the RBD, and how they can take advantage of this open conformation to design aptamers. Aptamers are a kind of is equivalent to an antibody, quotation, but made with nucleic acid in this case. So it's an oligo DNA, so DNA-based aptamer. So this is the oligo that they design, and actually you can see that in the models that they developed, it fits well to one part of the RBD of the central protein of the central domain that makes the recognition between the virus and the membrane cell. And actually, it is nice. They decided a different set of oligos, and you can see that they got a good affinity. This is, if you want to compare, this is the best that they get, and this is the best that was obtained by a massive in silico, a massive Celex type of experiment. So I think it was a big success to be able to design these short oligos that actually can, in principle, be used to block the interaction between RBD and AC2, so the income of the virus into the cell. The same target, different group, and this is a wonderful study, really wonderful study by Verdegrot and his group at MPI. And what they use here is a technology that they develop within BioExcel, and we are going to use later this technology to detect the changes in binding because of mutations in the interface. This is Rosetta. These are the results of the technology that they develop that is called PMX. So it's an accurate method to compute free energy. And what they did here was to mutate a series of residues. These are residues in the protein. But what, so, I mean, uh, you could expect that it's more interesting to look the mutations in the virus. They look the mutations at the protein because they want to design antibodies. So the idea is, okay, how can we use, in principle, one helix of the human AC2 to design something that binds so strong to the viral RBD that actually can interfere the binding to the human protein. So it will be an interfering antibody. And as you can see, these are still results that need to be validated experimentally. But as you can see, they found a small set of residues in which you can see some gain in stability in the complex. So things that in principle could help to design a better um, a better antibody, a modified peptide with an affinity for the RBD greater than the human protein. So in principle, you will be able to interfere in the binding. This is a similar type of study. In this case, they explore, we have done also this, but in this case, they explore what is the impact of mutations in the virus. It was at the beginning of the mutation, it was a big controversy in the literature. Um, you know, uh, People saying that there were mutations that increased dramatically the binding. What they found, and actually is the same that we are finding quite consistently, is that there are very few mutations that have a dramatic effect in binding. So there are very few changes in the virus that actually make the binder, makes the binding much, much better. So this is, there are of course, and you have the British variant, the Spanish variant, the Scottish variant, the blah, blah, blah variant, but actually the changes in binding are moderate, are in general, uh, it seems to be quite optimized. Let me use the now jump to, so I show you here examples of modeling study. I comment before some studies of method development. Let me show now one example in which we combine both. And this is in the context of one of BioExcel initiative. It's called BioExcel Building Blocks. And as you'll show later, is a coordinated efforts where computer scientists at BSc have had a crucial role. So what's the idea of the BioExcel building blocks? The idea is that, okay, we as a community have developed a series of tools, and then we can actually subdivide the tool in small pieces, in what we call building blocks. And then once you have done this exercise, you can combine in different ways the building blocks to make different workflows. Then we encapsulate these workflows in a workflow manager, and then in principle, if we do everything well, we'll have the possibility to run jobs with high efficiency, so everything is prepared for high performance computing environments, and that will be fully reconfigurable so that you can actually modify completely the workflow with a minimal effort. We were working for this for many years, and we had developed this kind of general framework. So we have the classical workflow structure with input-output files, intermediate files, and then we wrap up everything with this Python layer that, as you will know, is called PyComs. Is the the we say the language, no, the interface that Rosa Badia and her group has developed in the BSc. Okay, the method is very the workflow is very simple. So you say what you want to do. I don't know. I'm gonna download structures. I'm gonna model the things that are not there. Blah 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 blah. I will generate the topology, and then in this specific case, I'm gonna do mutations. So I'm gonna see what is the impact. So I'm going to run simulations changing arginine 5 into alanine, arginine 5 into lysine, arginine 5 into lysine. So very, very simple type of language where you can do then many things in a kind of automatic way without acting in every step of the process. So the wrapper is PyComs that allows a very efficient parallelization. I'm going to show you today results that were done in thousands and thousands of processors simultaneously. And this is how it works. So it takes a common, you take a common input, it divides into different nodes, and then as you can see, you obtain a very nice usage of the machine. So this is a real test, the first a real example. So the first input enter in the, April 25 at 10 o'clock at night. Then a few more processors became free. Then the rest of the jobs enter in the at 26 at midnight, and then you can see that you have a 100% coverage. So I would say it's perfect parallelism in jobs that, of course, have very little communication. Okay. So this is a technology that has been developed. Is wonderful to use high HPC centers, to use Praise, or to use Fenix, to use any big computer infrastructure. And we have this ready. We were using it for different purposes. When the COVID pandemic appears, we rewire everything. And this was an excellent example of how this type of, I would say, naive technology may be very useful, because it was just a question of a week that we were able to rewire everything and to launch COVID simulations. And this is what I'm gonna tell you today. So I'm gonna explain to you, but behind is the work of many people. I mean, the Berdegrot and Vitas in the MPI in Munchen, that they developed the basic physical method that is called PMX. Is for the experts in the field, is based on the Crookes theorem. So it's a free energy calculation method. These are the computer scientists, mainly, Rosa Badia and his group, Pau, Jose Luis group, and the people in my group that ran the simulations. Of course, Adam was also involved in he's a computer science. He was involved in setting up all the workflows. So you can see that what I'm going to explain to you now is the work of 11 people, most of them computer scientists, I must say. Okay, this is the workflow I'm going to comment. And yeah, it's you have some input files you generate for molecular dynamics, you generate topologies, you run calculations, you will run equilibrations, and then you run the PMX module that was developed within BioExcel. And this is a very clever module, because it follows free energy. It allows you to compute free energy differences between, for example, two mutants, but instead of the normal paradigm that runs equilibrium simulations and then are time dependent. So you need to run the same simulation in the processor for a long period of time. What it does is to run non-equilibrium trajectories. So instead of a long trajectory, it divides into hundreds of trajectories that they are collected in an independent way. And at the end, they are put together with this Crookes theorem that is a very solid physical framework. So making it sure, you run the non-equilibrium simulation in one direction, you run the same in the other direction, and these are non-equilibrium work. These are non-equilibrium work, but you can demonstrate that in the intersection, in this point, what you have is a reversible work. In other words, a free energy. This is how the workflow does. It runs simulations and then takes from these simulation seeds. From these seeds, it runs the PMX. It runs the non-equilibrium perturbation. So I will be very happy to give you any details if you want, but at the end of the day, what we have is a nice method to compute what is the change in free energy when you mutate this residue here to this residue here. And if you do this for the unbound state, for example, this is the RBD, this will be a mutation in the virus, then you do these mutations in the virus in the isolated RBD domain, or in the complex with RBD with AC2, you have these two free energy terms here that actually means absolutely nothing. But because they are thermodynamic measures, just subtracting this, you have an idea, or you have exactly the difference in binding because of the mutation. So what really interests you is the change in free energy of binding between this RBD that has the mutation and the wild-type RBD. And as you cannot compute this directly, you compute in this other way. At the end of the day, what you have is a delta G of binding, something that you can eventually compare with the experiment. Okay, I'm talking about a massive work, then we ask for support to Fenix or price support, and we were very grateful because we got the resources to do this, and then we start on a specific project. And this specific project was related to the entrance of the virus. So what you have here is AC2 is the receptor in the membrane of the cell. This is the RBD. RBD in this case is in what we call the one-up conformation. So this is a spike, sorry, and here you have a spike in the one-up conformation. So you have one RBD that is exposed. This RBD interact with AC2. After this, there is a kind of displacement of the entire protein here, and then the virus at the end of the day enters. So I'm not going to talk about further stages, but what is important is this first recognition step here. That is what you have here. We have X-ray structure, we have electron microscopy structure, so we more or less know how is the interaction. And then what we want to know is what was the trajectory of the virus before reaching humans. Because if we know how was these trajectories, we may know what will happen later, where the virus is going to go, and how it may affect again us in the 2, 3, 10, 20 years. So how the virus move from some species here into humans. And in particular, we know that the reservoir of this type of virus, of coronavirus, are bats. So how it moves from bats to human, and particularly to this special kind of bat that is called Rhinolopus affinis, how it arrives. And there is a lot of, newspaper conspiracy theories, whether it was the Chinese people or was Bill Gates or, very funny stories. But okay, assuming that all these stories are not true, how the hell the virus managed to jump from bat to humans? And the first thing that you can do actually is very simple, is to do this phylogenetic analysis where you compare, you can do this for the entire genome. It was already done in the first paper in Cell when the virus was characterized. And then you see that the closest thing that you have around is a virus from affinis, from Rhinolopus affinis, a virus that was found just in a systematic search. It's called RAG13. Is the closest analogs. But when you take a look to the RAG13 virus, you realize that at the recognition domain, at the RBD, the sequences are completely different. So I put you here, the RBD, this is the virus from bat, this is the virus from human, and what you have here are the dramatic changes that happen between one and the other. See that this is a quite small protein. You see the massive number of mutations and the massive number of dramatic mutations. Dramatic mutations mean mutations where you change the charge. That is the most aggressive that you can have. So virus RBD is completely different. But then you can take a look at what happens with the receptor to the AC2. Okay, if you think that the virus can jump from bat to human, you could expect that the AC2 of bat and humans were somehow similar. They are completely different. You see that there are more than 20 changes, and some of them absolutely dramatic changes. So you generate charges. So it's very different receptor, very different receptor binding domain, very different AC2. If you look now to the interfaces, and this is the software, the database I commented you before, you see that it's completely different. So this is RAG13. You see the interaction domain is completely different. The properties very different, also very different. The interface in the AC2, so in the cell protein. Very different between affinis, so the bat, and human. Very different from the affinis virus and the human virus. And then the conclusion is absolutely evident, is that in principle it should be a huge zoonotic barrier. Based on this, you will say, okay, this virus will never infect humans. And we as humans are always very, very, we found ourselves safe thinking in the zoonotic barrier, and this mysterious barrier that allow us to escape from infections from other organisms. So we use a technology I commend to you. So, and this is something very, very massive, very difficult type of simulations. But what we did was to make all the mutations between the virus in the bat, RAG13, and the virus in humans, SARS-CoV-2. And we also did the same mutations. We did this change in solution and in the complex. Okay, so from this cycle, you get an idea of what is the change in binding free energy in affinis when you mutate from the affinis virus to the human virus. And this is a negative number. And I must say that I was a little bit surprised because it says that the human virus is more effective infecting bat than the bat virus. But this is not so rare. There are many other cases where it happens. So apparently SARS-CoV-2 is an ultra-infective guy. It infects everything, including the virus. But so it's amazing. It's just curious, but it's not absolutely shocking. This number is shocking. What it shows here, this thermodynamic cycle, is that when you move from RAG13 to SARS-CoV-2 in human receptor, there is only a very small gain in affinity. So in other words, the bat virus could infect humans. And I put this face because I didn't trust this number. I put here the experiments. I'm not going to show you the experiments, but the experiments by Milan people that actually demonstrate that indeed this number is right, that there are not dramatic difference. There is not a dramatic difference between the human virus and the bat virus in terms of infecting humans. So think about to forget your ideas about zoonotic barrier. If zoonotic barrier exists, is not in the recognition domain, despite all the big difference. So we continue with this experiment. These are other thermodynamic cycles and also very difficult technically, but the numbers that you get here are quite amazing. So, or quite interesting, not amazing really, interesting. So this means, these numbers say you what happens when you change from affinis to human in terms of the RAG13 interaction. And you see it all in terms of the SARS-CoV-2 interaction. And what you see is that you have positive numbers, and these are beyond the errors that we can assume. So what it means is that probably everything was a virus evolution. So what these numbers suggest is that actually even the zoonotic barrier doesn't really exist in terms of the RBD-AC2 interaction, that probably this is a better host, that probably evolution happens in the virus and that eventually something closer to SARS-CoV-2 jump into human. This is a technicality, is an extra check of the accuracy of the numbers that we get by closing thermodynamic cycles with an error that in global was less than 1 kcal. So we can assume about 0.3, 0.4 kcal mole errors in our numbers. What is the theory that we have now? And this is completely work in progress, so I'm not sure about these numbers. So please don't distribute this number. They may be right, they may be wrong, but I think it's a reasonable theory. So if you take an alignment of all the virus, sorry, of all the affinis samples that have been collected, you see that they all are very similar, but in terms of the AC2, you can see that there is a majority sequence, and then there is another sequence where there are a couple of mutations. So in the colony of bats, there are apparently a few bats with a slightly different genome. And it's very interesting because those mutations are those that are found in humans. So our theory now is that perhaps there is a small part of affinis, not jumping to other bat, but just in the affinis community that has AC2 that is closer to the human one, and this was probably the intermediate step for jumping to human. Of course, this is just a theory, maybe wrong, numbers are still not converged. But as far as we know right now, the virus is an affinis issue. You don't need to call about any mysterious species in the middle. It may be just a lucky situation of a combination of affinis in the same ecosystem. This is a very recent paper saying that the last reservoir for the zoonotic barrier, that is what is called the furin site, is not anymore reliable because it has been seen how some bad coronavirus have taken this cleavage site spontaneously in evolution. So be ready for further zoonotic transmission. It seems that zoonotic barrier is a very, is a very nice concept, but it actually may not be true. And it may be possible really that we transfer our virus to other organisms, and sooner or later, these other organisms will return to us. We explore other things, for example, the impact of human polymorphism, actually making, a long story short, very small difference in terms of higher infectivity. There are some mutations that protect ourselves, but they have a very low prevalence, less than in the order of 1 every 100,000. So this is not going to protect us for infection. There are some that may increase a little bit our infectivity, but really the noise is larger than the number. Also, we are exploring, but I'm not going to talk on this, we are exploring virus variability. And actually, it's other groups have found the same, but very consistently, when you have a mutation that increases the binding between the RBD and the ACE2, you find that this mutation is selected by evolution. So they are variants of the virus that are successful, and they generate clones with the same genetic load. I'm not going to give you details on that because I'm running out of time. Let me just use my last slide to show you, as I told you many times, what I'm explaining here is only partially the result of our work. This is a massive effort for the entire Center of Excellence that actually we completely stopped the projects that we were doing at that time to jump onto this exciting problem, exciting and very serious problem of the COVID infection. So thanks a lot for your attention, and if you have questions, I would be very happy to try to answer them.
Say a few more words on the ICEI and Fenix services. So these are available to any researchers that are in need of scalable and interactive computing resources, virtual machine services, and archival or active data repositories. And you can have access to our resources through two calls. So there is the Human Brain Project eBrains call that this is ongoing, and it is dedicated to neuroscientists in Europe. And there's no deadline for this, as I said, it's an ongoing call. And then there are the Praise ICEI calls for proposals that Modesto used as well. These are quarterly, and Praise announces these, and we also announce them on the ICEI Fenix website. You can see here the link for the access to the resources, and you will also receive an email, a follow-up email after the webinar with all the links and all the details you might need for this. I also would like to ask you to give us your feedback, so that would be very useful for us to find out how you found our webinar today and what we can do to improve our training for the next time. So again, you will receive a link with this survey in this follow-up email. And just stay tuned with our updates and activities. You can join the Fenix User Forum. This is an online platform we use for Fenix users, and you can very easily become a member through our website. And this is our email, you can see it here, and you will also receive these details by email. You have our website here, and you can also follow us on Twitter. And with this, I think we can wrap up. I wanted to thank again Modesto for having this great presentation today, and BioExcel COE, of course, for their support, and all the attendees of today's seminar. So just stay tuned. You will be able to see the recording of this webinar and the presentation slides on our website very soon. Thank you.